Okay. So it's a great pleasure to me to introduce you to Dr. Igor Schapiro. Let me, this nice guy here. He's going to talk to us about understanding isomerization inside from molecular dynamics simulations. And just to have an idea about who is Igor, he got his degree in chemistry in 2004, and after that, he got his PhD only 10 years ago in 2010 in Germany, working with photoisomerization of retinal models. And after his PhD, he went to his first postdoc with Massimo Olivucci in the United States from 2009 to 2011. And then he returned to the Max Planck Institute, worked for another period of postdoc with Professor Lies, working with ORCA and other subjects. And then he stayed from 2011 to 2014. And then he moved to France for another postdoc, 1 year postdoc. And in 2015, he got a position in in the Fritz Haber Center for Molecular Dynamics in Jerusalem, where he is now. And a few days ago, he was promoted to associate professor in Fritz Haber Institute. So congratulations, Igor, for your new position. And
thanks.
For the main activities and awards, I need to emphasize some of them. And he got the postdoctoral research award in 2011. Another poster award in 2011 during the WATOC. And 2012, he got a postdoc award by IBM. It's called IBM Lovedin Postdoc Award. 2015, award for young scientist, and now very recent, 2019, Outstanding Junior Faculty Award in Computational Chemistry. The research interest that Igor is mainly working, involves apply and development of computational tools to understand chemical reactions. And he is, among the several applications he is working, he focuses on the light-induced reactions, in particular in chromophore protein complex. And he also interested in developing softwares, mainly computational tools with emphasis on multi-configurational wave function methods. And for MOCAS users, Igor is the one who implemented the track option in SLAPAF. So I think that most of MOCAS users are familiar with these options, track options in SLAPAF. So thanks, Igor, for implementing this option for us that makes our life much easier. And a few numbers, in 10 years after his PhD, he got around 60 publications, 5 chapters in collections, around 2,000 citations in Web of Science, and age index 20. So it's a great pleasure to us to listen to Igor today. And I thank you, Igor, for accepting these invitations. And Igor said that if you wish, you can interrupt him during the seminar. And I wish everybody enjoyed. It will be a great, great seminar, I'm sure. So thank you, Igor, for being with us today. And now I put the seminar in your hands, okay? Thank you very much, Igor.
Thanks a lot. Thank you. Thanks a lot, Antonio. It's a great pleasure to be here. Bom dia. Hope I pronounced it correctly.
Yes, bom dia. Very good.
It's the first time I'm giving a seminar in Brazil, and I wish I would be there in person, but unfortunately, due to the pandemic, we need to do it electronically.
Yes.
Okay.
There will be
Thank you very
other
much for
chance.
this detailed introduction. Yeah, I hope very much. So thank you very much for this detailed introduction. Now I want to, since most of the time I'm working with Zoom or WebEx, it's the first talk I'm giving using Google Meet. So just to double check, I'm going to ask you the most frequent question. Can you see my screen?
Yes, Igor. Yes.
Can you
We
also
can
see
see
the
it.
mouse pointer?
Yes.
Okay, perfect. Okay, so I'm going to talk to you about about one specific process that really fascinated me for a very long time, and it's the primary event in the vision process. So I will explain to you in detail in the introduction what it is about, and then I will introduce the molecule involved in this process, and then I will briefly mention the methodology that we use to study this process. And then I have, depending on how much time we have at the end, I have two sections of results. One is concerning the light activated, the photoisomerization, and the other one is about the night vision. So it's the mechanism which occurs in the absence of light. Okay, so let's start with the introduction to the vision process. Here on the bottom, you see a slice of a human eye. Here in front, we have the lens, and on the back side of the eye, there is a layer shown in orange. This is the so-called retina. If we zoom in into the retina, which is shown here by this photograph on top, you see that it's composed of two types of cells. There are these elongated cells, which are called rods, because they have a shape of a rod. And then there are slightly shorter cells, which have a shape of a cone. And that's why they carry these names. Now, they contain a protein which is light sensitive. So when light is entering through the eye, it goes through the lens, and then the lens focuses the incoming light on the retina. And actually, if you look at this scheme, because of the lens, what we actually receive the picture upside down. It is our brain which is correcting for this and returning the picture to its normal orientation. Okay, but now my focus is more on the pigments inside the rods and cone cells. So in one eye we have 90 million rod cells, and we have 4.5 million of so-called cone cells. Now inside the rod cells, you see we have much more of them, 20 times more rod cells. We have a pigment which absorbs which has an absorption maximum at 500 nanometers, and it's this spectrum shown by the dashed line. Now from cone cells we have three different pigments, and they absorb short, medium, and long wavelengths, and this corresponds to red, green, and blue. So from here it's clear that the rod cells are just responsible for monochromatic vision, which is the so-called dim light vision. So it's the vision which is working at night, because as you know, in very dark conditions, in the absence of light in the evening, you actually cannot distinguish colors. You see everything monochromatically. This is called dim light vision. Now the cone cells, because there are much fewer of them, they are not operating at night, but they have the possibility to distinguish colors. So they are responsible for color vision. Now the work that I'm going to present you is mainly concerned with the pigment from the rod cells. Here is the schematic display of a rod cell. It has this elongated antenna-like structure, and in the top of the antenna you have a high concentration of so-called discs. Now inside the disc, well actually if you look inside one of those discs, they have a membrane which separates them from the outside of the cell, and inside the membrane, so in this light blue layer, there is a transmembrane protein. So the protein is going through the membrane. It has parts sticking out in the extracellular side and in the inside the cell, and it's characterized by having 7 transmembrane helices. And these helices, they form some sort of a barrel. And inside the barrel, you see here, there is this red molecule. I mean, in the scheme, it's red. This molecule is an organic molecule. It comes originally from carotenoids. So, and it's the precursor of vitamin A. So vitamin A is predominantly found in carrots, it's found in apple, and that's why it's good for the vision, because this molecule is then getting oxidized and goes to our visual cells, and then it's incorporated inside rhodopsin. So when it's incorporated inside rhodopsin, this aldehyde forms a Schiff-based bond with the lysine. Lysine is a side chain of this protein, and then it becomes covalently bound. So there is a chemical bond which keeps the retinal inside the protein. Now, why does it absorb in the visible light? Because it has an extended conjugated system. It has 6 double bonds, 1, 2, 3, 4, 5, 6. And they are conjugated, and all of them are trans except the one in the middle, the one connecting carbon 11 and carbon 12. This is the double bond which is in cis configuration. Now, the primary event is when light enters through the lens and is then absorbed by retinal in rhodopsin. And upon absorption, this 11-cis double bond is going to all-trans. Now, this reaction has unique characteristics. It's completed on the scale of 200 femtoseconds. This is femtosecond is 10 to the power of minus 15. So it's 15 zeros. It's extremely fast. It has one of the highest yields for photochemical reaction in nature is 67%. So it means 2/3 of the molecules go successfully to trans, and 1/3 returns to cis. And the most relevant for chemists is that from all the 6 double bonds, only one is selected. There is nearly 100% bond selectivity. And this is kind of a dream reaction for chemists, because they want to have selectivity and specificity. And this is somehow achieved in the protein, because outside of the protein, for example, in ethanol solution or methanol solution, this retinal protonated Schiff base does not have this characteristic. So the isomerization time or the excited state lifetime is 1 or 2 orders of magnitude larger, so it's somewhere in the picosecond region. The quantum yield goes from 67 to 20 or even below. And instead of one isomer, we have a mixture of different isomers. So somehow protein is catalyzing this reaction, and so that's why already since the discovery of retinal and rhodopsin, people have asked the question, what is the mechanism? How does it work? Now, if you go to a textbook in organic chemistry, then what you will find there is classical one bond flip. So it means that on the paper it's written like you have 2 ends connected by double bond, and then one of them is fixed and the other one rotates by 180 degrees. But inside protein, there is not enough space. You cannot make this large motion because the molecule is going to bump into the protein environment. Moreover, this chromophore retinal is sitting very tightly in a pocket. So there must be another mechanism. Now, in 1976, Aryeh Warshel, who is one of the pioneers in computational chemistry, he came up with the so-called bicycle pedal mechanism. What he said is that, I hope you can see it on your screens, that 2 double bonds which are next to each other, is the one marked in black and the one here next to it with the arrow, they rotate at the same time. And the motion, I'm going to show it with my hands, I'm not sure if you can see me, but the motion is like rotating pedals in a bicycle. And that's why he called it the bicycle pedal mechanism. So he basically says that two double bonds isomerize at the same time. Now, this was really a pioneering effort. You know, in 1976, there were no personal computers. He asked NASA to give him time on their supercomputer center, which was quite primitive at that time. There was also no structure of the protein, so he modeled the protein environment using van der Waals spheres, and he also fixed the two ends of retinol. Nevertheless, despite all these approximations, it was the only model that was able to explain conversion between different forms of retinol, meaning going from rhodopsin with 11 cis retinol to all trans and also to 9 cis. But a few years later, in the 80s, there was another group from Robert Leo, who is based in Hawaii. They came up, probably inspired by local activities, they came up with the hula twist mechanism. So in the hula twist, there is, instead of two double bonds, there is a simultaneous isomerization of a double bond and a single bond. So here you see the highlighted bonds. There is this double bond from 11-12, and next to it there is the carbon 10, carbon 11. This is a single bond. They isomerize at the same time according to the hula twist mechanism. And the reason why this hula twist mechanism was superior to the bicycle pedal is that, according to the authors of the paper, hula twist requires even less volume inside the pocket. So it means that it should be, I mean, according to the authors, it should be preferential. However, there is one problem. It doesn't result in all trans. So if you isomerize a single bond and double bond at the same time, it will give you 10s, which means single, cis. So it was contradicting the experimental results, which was found later. Yeah, nevertheless, it was didn't stop the authors from publishing papers until 2000. So a few years later, already in 2007, the group of Massimo Olivucci came up with a crankshaft mechanism. In the crankshaft mechanism, there was not only one, two, or two double bonds involved, but multiple. All the double bonds along the polyene chain, along the conjugated chain of retinol, were rotating. Similar to this model of the crankshaft. And because of the rotation of multiple bonds, so like all of them twisted a little bit, you see the twist is 45 or minus 35 degrees. It allowed for a space-saving mechanism. Then another proposition was made 2 years later by the group of Hans Lischka, together with another Brazilian fellow, Mario Barbati. They came up with the folding table mechanism. So, and according to their picture, I've seen this figure already many times, but I'm still struggling to see where the motion resembles the folding table. But according to their model, there is a simultaneous isomerization of 3 double bonds. And this is here the figure with the caption E. So this is the central double bond, which is rotating in one sense, and then you have two double bonds in the periphery next to it, and they rotate in opposite sense. So this mechanism was proposing that 3 double bonds are involved in the isomerization. And then, last but not least, in 2012, the group from Ishida in Japan, they came up with the wing and wet towel motion. So what they propose is that you have a simultaneous twist along the whole polyene chain, similar to the motion that you do when you want to squeeze out the water from a towel. And this somehow resembles, in my opinion, the crankshaft mechanism proposed by Olivucci group. Well, nevertheless, we decided to study this process using ab initio QMM molecular dynamics. And before I go to the results, I wanted to say that rhodopsins are not only involved in vision, they are present everywhere. So we have a related protein called melanopsin, which is found in the brain. So it's not in the eye, it sits in the brain. And this this protein is regulating our sleeping and awake cycle. This protein also absorbs blue light. So that's why when you are when you're watching at your phone at night in the evening, you activate this melanopsin protein, and this perturbs your sleeping cycle. So that's why I think this is one of the reasons why blue light filters were introduced to computer and phones. In flies, like this Drosophila example here, rhodopsins are involved in thermal sensing and hearing process. And last but not least, there is a whole new field in neuroscience, and it's called optogenetics. In optogenetics, a channel rhodopsin, so it's a rhodopsin which when it's activated by light, it starts to channel ions in and or outside the cell. This channel rhodopsin was used in very early studies by Karl Dysenroth and Peter Hegemann from Germany, was used to control the motion of the mouse. So there is this famous video where the mouse is randomly walking in a cage. When you shine light, it triggers the mouse to run in circles. And when you switch off the light, the mouse stops. So it allows you, channel rhodopsin allows you to control living organisms just by light. So, but then I hope that this introduction, motivated you, and then you see this rhodopsin presentation, and you go after the presentation to the protein database, where you can get crystal structures as a starting point for simulations. You will find immediately that rhodopsin has 5,521 atoms, and this is without adding the hydrogens. So this is clearly too large for quantum chemical simulation, as you know it from the work of, for example, Antonio in your institute. So 5,000 atoms is there's no way you can do it, and at a reasonable level. In order to overcome this bottleneck, these three gentlemen proposed already in 1976. They proposed a multi-scale model. Actually, it was Michael Levitt and Arya Warshel. Martin Karplus came up. I mean, his contribution was slightly later. So it took almost 40 years to recognize the importance of these developments, and they received the Nobel Prize in Chemistry. So what they have done is something called multi-scale modeling. And let me explain you this concept of multi-scaling. So here you see the photograph of Alfred Nobel, the founder of the Nobel Prize. He invented TNT, and he donated his money to establish the Nobel Foundation. So you see his photograph is divided in different zones. Now, what do we need to recognize a person? Well, the most characteristic of a person is the face. So you see his face is in the central zone. This zone is highlighted by a white circle, and it has the highest resolution. So here we can recognize every little detail. If we move outside of this zone, if we move outside this circle, we move to a second zone, and here everything is pixelated. So we have reduced the resolution, but the reduction still allows us to see that he is wearing a white shirt and a black suit. So it's still enough to recognize details. Now, if you move from the second zone to the third one, here is the point where we are losing details. Everything is just blurry and averaged out. Now, the same concept can be transferred to an enzyme or a photoreceptor protein, because we don't need to describe the whole thing at the highest resolution. The action happens in the center. We have an active site where electrons are used to form bonds, break bonds, or where electrons are excited. Here we need the highest resolution, like in the face, because the active site or the chromophore is the face of the protein. And here our resolution is that we have electrons and we have nuclei. If we move out of this central active site to the protein environment, here we don't need to describe bonds which are breaking. We don't need to describe any situation where electrons are required. So here we can reduce the resolution. We can omit electrons, and instead use a cheaper method. When I say cheaper, I mean computationally cheaper. This is, for example, a classical force field. Now, in the classical force field, we don't have electrons. We have just parameters, and they are put in explicit equations for bonding and non-bonding interactions. So just to give you an idea, to calculate the active site, we need a few hours at the QM level, but to calculate a much larger protein, we just need a few seconds. And then similar to this third zone, which is just completely blurry and completely average, we have also in our toolbox, we have also the possibility to use a dielectric constant, which just omits completely any atomistic resolution. Okay, so now this is so-called multi-scale approach for simulation. Now, if you're talking about rhodopsin, then it becomes clear retinal is the one that is the part which is light sensitive, so it needs to be in the QM region, and the rest of the protein is the classical part described by protein. So the division between them is very clear. However, as I mentioned in the beginning, retinal is covalently bound to the protein. So at some point we need to truncate a bond. There is no way we can separate them from each other otherwise. So if we want to truncate the bond, we need to cut it. And then if we cut a bond in the middle, homolytically we get a radical. And a radical has very different properties. So this boundary between the QM and MM region, between the high resolution and low resolution, is extremely problematic and extremely sensitive. However, nowadays we know how to deal with it. There is something called the link atom scheme, where we cap the truncated bond with a hydrogen, and we put this hydrogen just between the two carbons. This is the way to deal with it. Okay, so this is the way how we obtain the energy, by using this multi-scale model, also called QMMM. Or it's better to say that QMMM is a special case in multi-scale modeling. But now, how do we move on the surface? How do we study a reaction? Well, the best way to do it is to use molecular dynamics. Now, how does it work? Here is a potential energy surface, and you have to define your starting point. So a starting point is just a dot on the potential energy surface, and that defines the start of your simulation. This is time 0. And then what you do is, for these coordinates on the potential energy surface, you can use any computational method to get the potential energy. So here I'm showing you, this is the expectation value from this Hamiltonian. So it's some kind of quantum chemical method that gives you the potential energy V. And then once we have the energy, we can form the derivative. This can be done numerically or analytically, depending on the method. And the negative derivative is the force that is acting on these coordinates at time 0. Now, when we have the force, the Newton force, we can extract from it the acceleration, because the force is just the mass times the acceleration. And then, once we get the acceleration, if we multiply it with the time step delta t, we can get the change in the velocities. So this completes the cycle. We are going to get now from one point, from the starting point to the first point, and then we repeat it again, and so on. And this is how we propagate in time. So when we choose the time step small enough, then as I showed you here, then we can just connect the separately obtained snapshots and make a movie out of it. So at the end, we get a smooth motion of the molecule. Now, in order to do it, what I have done is I implemented the molecular dynamics model and the surface hopping model into MolCAS, and also with the help of the collaborator in Marseille, Nicolas Ferret, we also extended it to QMMM by using his ESPF model. So here's a typical workflow, and I guess many of you are familiar with MolCAS. So you have first CUR to calculate the integrals, then you put the QMMM part, ESPF. Here's the part which gives you the CASSCF wave function. Then we have the Alaska MCLR. Here we are going to solve the coupled perturbed CASSCF equation to get the gradients and the state-specific correction. And then in RASI, we calculate the overlap of these different CI vectors. So we calculate overlap of different wave functions for different steps. And then from there, we get input for surface hopping. So we decide in the surface hopping whether to go down to the state or go up. So these are really details now. And then we have the dynamics model. Recently, Molecules decided to split the development, and now there is an open source version called Open Molecules, which everyone can download from GitLab, and still at the same time there is a commercial version which continues to be available, and it's actually has almost the same capability. I mean, it says virtually the same capabilities. In fact, nowadays, I think Open Molecules is more has more features, or is more updated. Okay, now with this implementation, I was able to study the photoisomerization in rhodopsin, and I want to show you. I want to start in this first part by showing you directly the results. So here, for clarity, I removed the protein environment. I am showing you only the retinal. And the orientation of the retinal is as shown here in this topper. In this top Lewis structure, you have the Schiff base. Sorry, on the right side, you have the beta union ring, which is shown here. And here on the top left, you have the Schiff base, which is the bond, double bond to the lysine. And you see here, there is a little bit of blue indicating on the nitrogen, which is on the top left. Okay, so now this is the simulation which starts in the excited state, but then propagates in time using molecular dynamics and ends up in the ground state, hopefully in the correct isomer. So when I press play, the molecule is going to get excited. Okay, but I forgot to mention that in addition to the geometry, so what you see here in gray, I also show the pi electron density. So this is the red part here. And these are not orbitals. This is density. So it's the square of the sum of all the pi type orbitals. And you can clearly, why is it useful? You can clearly see where we have a double bond. Wherever we have a density between two atoms, this is a double bond. Like this is the carbon 11, this is carbon 12, and they have a double bond. Now if you go to the next pair, this is a single bond because here we have a knot. Then we go to the next one. This is double bond, single, double, and so on. Now when I'm going to press play, we will excite the system. And I'm doing it right now. You see immediately after excitation, the double bonds are broken. Now everything is shifted. The density is shifted by one atom. And now since we have a broken bond between these two carbons, we can see a rotation. And at the end of the simulation, which we're coming now, you see that the density is restored. It's again a double bond, but now these two hydrogen atoms are on opposite sides. So this is a trans conformation. But at the beginning, at time 0, they were on the same side. So this was clearly cis. So you saw a successful isomerization of from cis to trans. And now I want to go with you slowly through this analysis of the trajectory. So I showed you the movie, which is based on this simulation. And I also have saved the data, so I have the energetic evolution, the evolution in energy. So it's for the simulation I showed you from time 0 to time 200 in femtoseconds. You see the red potential is the excited state, the black one is the ground state. What happens at time 0 is that you have a maximum gap. You excite with the photon with this energy. You move on the excited state. You move, and then the gap is getting smaller and smaller. And at some point, which is shown here by this dotted line, you have a transition between the excited state and the ground state. Now, this transition is the time where we are passing through or close by to a conical intersection. We don't know exactly, but the energy gap is here very, very small. And from this time, we're continuing the simulation on the ground state potential energy surface. So if you just form the difference between the red and the black, you get the blue delta E curve. And here, you see we are going, the energy gap is decreasing sharply. In the first 15 femtoseconds, it's going down by a lot. And then it steadily decreases at a slower pace. And then here becomes almost zero. This is the point we saw already here. It's not exactly zero, but it's very close. And then it increases again. So for us as chemists, we want to know what is the mechanism. And this was also my question at the beginning. What happens here? Why is this, has this evolution, is this characteristic steps? Now, if you look at the geometry, on the right side, you see now graph panels describing the geometry. And here I want to remind you, this is the retinol. So let's look first on top, where you see the bond lengths. The two lines in red are marking two double bonds, 11, 12, 13, 14. The two blue lines are highlighting the single bond, 12, 13, and 14, 15. So let's read this graph. At the beginning, at time 0, the double bond is short. The single bond is much longer, as it should, as expected. But if you remember the movie, at the beginning when I start the simulation and I excite with light, then double bonds were broken and the single bonds become double bond. I mean, they got a pi contribution. And this is exactly what we see here. Because the electronic structure in the excited state has changed, there is an intramolecular charge transfer inside retinol. The bonds are rearranging. So the electronic structure changed instantaneously, but the nuclei require some time to change. And this takes place on a scale of 15 femtoseconds. Here all the single bonds become very short, and the double bonds become elongated. And this time is in coincidence with the time that we saw here. The drop in energy, it's more than half of the energy, okay, between the ground and excited state. So it's this point here. This is changing on the same time scale. So it means the first coordinate is the change in the so-called bond length alternation, when double bonds become single bonds and vice versa. This is responsible for this very fast and quick drop in energy. Now you see, on the excited state, the double and single bonds, they oscillate around the constant value. Okay? But once we return to the ground state, then there is again a transition period, and then again it becomes stable. So the single bond becomes longer, double bond becomes shorter. But what happens in between? Well, we can look at this evolution of the Hedel angle. The black line corresponds to the Hedel angle defined by these four atoms. 1, 2, 3, 4. The red line is the one describing the same double bond, but it's defined using 2 hydrogen atoms instead of 2 carbons. And hoop is so-called hydrogen out of plane mode. So there's the difference between these two curves. So now, at time 0, this dihedral is not changing. Sorry, in the first, I meant to say in the first 10 to 50 femtoseconds, this curve remains virtually flat. But here, once the double bond becomes stretched, okay, once it's stretched like this, then there is enough space for the double bond to rotate. Now, the hydrogens can move out of the plane. And this you see here, there is an onset around 15, where the angle starts to, the Hedel angle starts to increase, and finally it reaches 90 degrees, or almost 90 degrees. This is the time of the transition to the ground state. And now it continues already in the ground state and forms the product which has 180 degrees, or close to 180. Okay, so now with this information in mind, let's have a look at the movie again. So these are the double bonds, 1, 2, 3, 4, 5, 6. I'm going to press play, and you see these double bonds immediately break. They're becoming elongated. They are single bonds. And now you see that there's an onset. This hydrogen moves out of the plane. This one stays in the plane, but then it follows with a little bit of delay. And now we have already the formation of the double bonds in a new conformation, namely trans. So this is the mechanism, and I explained to you the critical role of the electronic structure, the double bonds, and the dihedral angles. So first we have a change in the bond length, which is called bond length alternation, and then we have the twist and isomerization. Now at the beginning I showed you several different mechanisms, so the question is, what mechanisms do we observe here? If I press play again, you see all of the motion is done here in the center, but there is also a large motion here of this metal group. You see at the beginning it was sticking out of the plane, pointing towards us. Now it goes inside the plane. So that's why I believe that it resembles closely the model originally proposed by Arya Warshall, where two double bonds are isomerized in the so-called bicycle pedal mechanism. However, this bond does not fully isomerize. So instead of coming up with a new name, we call it an aborted bicycle pedal, because the second double bond does not fully isomerize. Okay, so we explained with this study, we were able to explain the mechanism and what happens during the isomerization. But now, what can we do with this information? Well, we can look at the time of the transition. So it's around 100 femtoseconds, where we have the transition between the excited state and the ground state. And I mentioned that the dihedral angle is roughly 90-degree twisted. So what happens at this 90-degree twisted? So here you see a Newman projection along the carbon-11, carbon-12 bond. So in front is carbon-11, and behind, you cannot see it, it's carbon-12. And this is the worst situation for a double bond, because the two p orbitals, which contribute to the pi bond, they are perpendicular to each other. And so there is the smallest overlap. Now, what happens right after this crossing? Then there's one scenario where these two hydrogens, shown here and here, they move away from each other. They move away, and this leads to an increase of the overlap of the lobe B from the p orbital and lobe B prime. And this increased overlap leads to formation of a double bond and eventually to the trans isomer. In the other scenario, the hydrogen atoms are moving towards each other. Now this leads to an increased overlap between B and A prime, as well as B prime and A. So this initial motion makes this overlap increase, and it establishes the Z isomer, or sorry, in other words, the cis isomer. So in nature, the ratio is roughly 65 or 67 to 35. So in 2/3 of the cases, from the conical intersection we go to the trans, and in 1/3 of the cases we go back to cis. So this is, of course, a purely computational study. But when we came up with it, and we mentioned that hoop this hydrogen out of plane mode, I'm talking about these two hydrogen, that it has a critical role and it's considered to be a reaction coordinate, then a group in Berkeley, a spectroscopy group, decided to test it experimentally. So what they did is they prepared a sample where these two hydrogen atoms are replaced by deuterium. So only the hydrogen atom at carbon 11 and carbon 10, sorry, carbon 11 and carbon 12, only these two were replaced. And they synthesized, or sorry, they tested 3 different isotopomers. In one case, hydrogen at position 11 was replaced by deuterium. In the second case, hydrogen at position 12. And in the third case, both hydrogens were replaced by deuterium. Now, if they replace both by deuterium, then actually nothing changes in the ratio. The ratio of 2/3 to 1/3 remains almost the same. However, now, if they replace only one of them, then this symmetric hydrogen out of plane mode becomes asymmetric because one of the hydrogens now has double the mass. So we are breaking this hydrogen out of plane mode, we're making it asymmetric, and this leads to a large perturbation in this stereoselectivity. So it means that now, according to the experiment, 55% were forming cis and 45% forming trans. So it basically confirmed our finding from simulation that the hoop is a very sensitive reaction coordinate that can be used to alter the reaction. Okay, I see that I'm actually I used up all the time. So we have almost 5 o'clock. I have a second part, but I think maybe it's going to be a little bit too much. I think there are five slides. So if you want, we can make questions now. We can have a question session. Or if you really insist, I can also continue.
Okay, Igor, go on.
Okay. Okay, good, thanks. But okay, want to ask questions at the end, or okay, I will just go. I can.
If you have questions, you can ask now. Otherwise, Igor goes on.
Okay. I personally think this is the most exciting part. I think this is the most interesting contribution. One of the most interesting contributions because we were using quantum chemistry and we were able to explain a physiological process. So let me explain to you what I mean. So this is the rod cell that we studied the pigment inside the rod cell, which is called rhodopsin. So there is a group, or there are several groups in the world who can extract a single rod cell from the eye and measure the electrical signal coming out. So when you shine light, here are the rhodopsins. They undergo something called a photoreaction, and at the end it produces an electrical signal inside the membrane, which leads to the synaptic end where the cell is connected to a nerve which goes to the brain. And they're able to measure the electrical signal. This electrical signal from a single cell is something on a scale of picoampere. And you see this, there is a lot of noise, and then from time to time there is one peak coming out. So there is some electrical signal produced when you shine light to a rod cell. But then when they were measuring this, when they were doing these experiments, they noticed that in the total absence of light, without any light, they still have these peaks. So there are from time to time these peaks coming from showing an electric signal, but there is no light. It's in complete darkness. So these findings were attributed to a thermal, instead of the light activated, isomerization. So they call it the thermal isomerization. And they showed that the thermal signal is exactly the same as the one from the photoactivated. However, there is one little problem. The barrier for the thermal activation is so high that in principle we should see one of this isomerization happening in once in 400 years. So it's a very, very rare event. However, I told you at the beginning that we have 19 million rod cells in one eye. And each rod cell has 2 billion rhodopsins. So we have such a huge number of rhodopsins that it happens every few minutes or every few seconds. And actually, this was known already for many years, that we have some kind of thermal noise which is decreasing our visual sensitivity. So at very low light conditions in absolute darkness, there is this noise which was first discovered by Germans and called Eigengrau, which means intrinsic gray. And it was described as a uniform dark gray background that many people report seeing in absence of light. So instead of being completely black, it's something kind of very dark, and from time to time you see like little dots. Now, there was another scientist, Sir Horace Barlow, who was a professor in Cambridge. He measured rod cells from different organisms, and he found a correlation. So he found that there is a correlation between the wavelengths. So I have to say that the difference between different organisms lies in the composition of the protein. So there are some amino acids replaced with some other, and this leads to a different, this polarizes the retinal in a different way, and that's why the absorbing wavelengths or the absorption maximum is changed. So he found a correlation between the absorbing wavelengths, which is here on the x-axis. So it's actually 1 over lambda max, the absorption maximum. So this is linear in energy. And on the y-axis, you have the negative logarithm of k, which is the reaction rate of the thermal isomerization. So he found that the more blue shifted the absorption, the less thermal noise he was observing. And this was established in 1957. However, it was not clear what is the reason for this correlation. So the more blue the absorption of the visual pigment, the less prone it is to thermal isomerization. So let's try to explain what it means. Yeah, this is the explanation. So moving to the right, it means to the blue absorption. Moving to the left, becoming more red. And moving here on to the top means the thermal noise is low. Moving to the bottom means it's high. So let's again summarize what I just told you about the photoisomerization. You were starting in the ground state. There is a photon of light. You promote the molecule to the Franconin region. Then this excitation is characterized by the lambda max value, the absorption maximum. And then you undergo a photochemical isomerization. So there is the movement here. We pass through a conic intersection, and then we reach the product. Now, what I didn't tell you explicitly, I mentioned that there is some intramolecular charge transfer, but I didn't explain it in detail. So when you excite retinal, you have here these charges. They are represented by spheres. A small sphere means small charge, big sphere means big charge. And blue means positive, red means negative. So we have this charge distribution in the ground state, but once you excite, there is a partial charge moving from the beta ion to the Schiff base, or if you talk about positive charge, it moves from the Schiff base to the other part, the beta ion part of the retinal. And this is demonstrated here. Charge was sitting before here, now it's sitting here. Okay, this is the photoisomerization. Now, what about the thermal isomerization? The thermal isomerization is occurring in the ground state. And in order to occur, we need to break the double bond. So we need to overcome a thermal barrier, which is characterized by transition state with a well-defined activation energy. So instead of proceeding through the excited state, it proceeds in a ground state. Now, the correlation from Barlow was relating the lambda max value here and the activation energy shown here on the y-axis. So we need to find a connection between the excitation energy and the activation energy for the thermal process. So now, just for those maybe who heard about conical intersection but they don't know exactly what it means, I would like to illustrate is that transition state, a concept you're all familiar with in a ground state, is describing a stationary point which allows to describe a reaction from A to B. So on the way from A to B, there is a transition state, and this transition state describes how to go from A to B, and there is one vector which tells you the direction to go to the product. Now, conical intersection has a similar meaning for the photochemistry like the transition state for thermal chemistry. So it's a very ubiquitous, very common feature in photochemistry. However, like the details of a conical intersection are very different from a transition state. A conical intersection is not a stationary point, it's a singularity. It means that the derivative at the conical intersection is not defined. Now, instead of having one transition vector, there is a branching plane which is defined by two vectors. And these two vectors describe how to leave the degeneracy, how to escape this conical intersection. And finally, conical intersection can lead to more than one product. So if you look at this example, at this scheme for rhodopsin, then we have rhodopsin, and we can go from rhodopsin to the all trans, which is called the beta rhodopsin, and we can pass from the reactant to the product on one side of the conical intersection, and we can pass to it also on the other side of the conical intersection. So there are two pathways to reach this product, okay, and they require that you pass the conical intersection from different sides. Now, in 1963, Herzberg and along with Higgins, they showed that the electronic functions for two states crossing at the conical intersection undergo a large change when moving a molecular structure along a loop which encircles the conical intersection. So it means if you rotate around the conical intersection, the wave function is changing. So what it means practically for us is that one of the points, transition state here, okay, so when you pass from the reactant to the product, you have a transition state. And one of them has a diradical wave function, the other one has a charge transfer. And it's not only in this point, but it's in the whole region shown here in brown. So let's explain it in terms of chemistry. It means when we go from cis to trans, we are passing through the point of 90-degree twist. Now, this 90 degrees, like this, the two bonds, it can be reached by breaking the double bond homolytically. Means that we have a radical system, a diradical, hence the name diradical wave function, diradical transition state. And we can have a full transfer of the electron pair on one side or the other. So it means that they will have different polarizability. They will have different response to the protein, because in one case you have a full transfer of an electron pair, in the other case you have a diradical. And what we also found is that the charge transfer transition state is 11 kcal per mole lower in energy than the diradical. So this is the preferred way, and this is the exclusive way. Okay, so now what happens in this experiment from Horrens-Barlow when you check different mutations or different organisms? So when you have now retinal, you see these are the charge distribution in retinal, and let's say you introduce a negative charge. So, and here I forgot to mention that we have the ground state, we have the excited state, this is the excitation energy, this is the transition state, and they can be in a diabatic picture described by two diabatic states. One is the theoretical state that I showed you, and the other one is the charge transfer. So when you introduce a charge through a mutation, this is just an example, okay, to show you the different polarizability. Because the charge transfer and the theoretical have different charge distributions, this charge is going to have a different effect on one potential compared to the other. So meaning this, for example, charge transfer state can be lowered, and this lowering will have an effect on the excitation because the excitation energy is lower now, but at the same time the transition state is also lower. And this explains the link that Hornet-Barlow has found. So in order to further extend this study, we simulated the Barlow correlation for 5 mutants of rhodopsins. And then we correlated the charge transfer transition state and the radical transition state. The charge transfer transition state gave us this slope here shown in red, while the blue one was obtained for the radical. And you can clearly see that only the charge transfer is giving us for blue shift a higher thermal barrier, so it means this is the one that gives you the Barlow correlation, while the radical has a complete opposite behavior. Now, from our simulation, we can also we can also see that at some point, when you continue further in the blue, at some point, there will be a crossing between these two lines. And then, instead of the charge transfer, the theoretical transition state will be preferred. So we are very close here to the optimal. This is the human vision induced by rhodopsin is somewhere here in the very top. We have a very small thermal barrier, so we can see very well at night. And at some point, we know that if you push the absorptions further to the blue, by other mutations, you can have an inversion. But so far, we are missing the experimental evidence. Okay. Yeah, so with this, I'm coming to the end. This is the acknowledgement slide. And here's the list of coworkers and group members who I would like to thank. And also thank you for your attention. And I will be happy to take questions.
Thank you very much, Igor, for your nice talk.
Welcome.
So, questions, please. You can ask via chat or using the microphone. Any questions?
I see Sebastian also came. Hi, Sebastian.
Sebastian, yes, Sebastian is.
I have a question.
Hi. Yes, there's a question.
Can you hear me?
Yes, very good.
Okay. In the first example about the isomerization of retinal, you showed the time evolution of some single and the double bonds, right?
Yes.
Instead of that, you could define a bond length alternation, which is the difference between. Okay. My question is, have you ever taken a look on the bond order alternation instead of bond length alternation?
Yes, that's a good question. Yeah, actually, in a more recent work where when we compare different mutants, indeed we looked in bond order. We computed the bond order instead of alternation, and it was actually a better. It was giving it as a better correlation with the results. So yeah, we found that, as you say, the bond order is a better description than bond lengths.
Okay, thank you.
Igor, in the first part,
yes.
when you showed us some trajectories, how many trajectories do you compute?
Yeah, yeah, this is also a good question. Well, in this original study that we did, at the beginning we computed, I think, 30 trajectories, which was taking up a lot of time. But as you know, now the hardware advanced and, molecules advanced. Now we have Cholesky decomposition, and they are also much faster programs. So now we are running routinely 100 trajectories. But yeah,
100.
the analysis that we are running, we have hundreds of, I mean, the smallest ensemble is like 100. We are trying to do more. But so the trajectories that I showed you was basically one representative trajectories that we studied for the mechanism. So we checked it also with other trajectories, and like it showed very similar behavior, and it followed the same mechanism.
Okay.
One, Igor.
Yes.
Of course.
But yeah, I mean, also thanks for the talk, and was quite nice. What kind of non-adiabatic coupling representation are you using in Volcas? Is this just non-adiabatic coupling vectors from Alaska or overlaps from Rassi? And
yeah, in this specific, in the studies that I showed you, which was from earlier work, we're using the overlap from CI vectors in Rassi.
Okay, and that is taking into account how? Is this, do you make numerical derivatives of the overlaps, or is it this local diabatization scheme or something like that?
No, so we are in this study that I showed, what we are calculating the overlap of CI vectors, and it gives us a value between 0 and 1. And then we have, instead of the stochastic tally surface hopping, we're using a diabatic surface hopping. And then we have some fixed values. So if the overlap deviates by, I think by a quarter from the ideal overlap from orthogonal wave functions, then we induce a hop. So it's a deterministic hop.
Okay. And that works well?
Yeah. So in this dynamics model, Alessio from who was a PhD student with Massimo and also with Luis Manuel Frutos in Madrid or in Alcalá, they implemented also surface hopping, and they implemented tally surface hopping, and we compared the results, and it was giving basically the same numbers. It was giving the same mechanism and the same reaction mechanism. I guess it depends. It depends on. I guess it depends on the problem. So in rhodopsin we have really a very ballistic transition. It goes just down the potential, and there's always a transition very fast. But if you have weakly coupled potentials, maybe you need slightly different schemes.
Yeah, that's what I would also expect, that it works well because you have this peaked conical intersection where there is only one choice. Okay. Yeah. Thanks.
Thank you.
Any other questions? No other question? Okay, so I just want to thank you again, Igor. Your seminar was really
Antonio,
nice.
thank you for the invitation. Thanks.
No, you are welcome. And I hope to see you, Sebastian, Felix, in Brazil soon. So we can have a presidential conference or things like that. It will be very nice.
Happy to come.
Let's see when.
I will be happy to come.
Yeah, let's see. Okay, and if you have questions, please email Igor, and he will
I forgot to show my email.
So thank you very much, Igor, and I hope to have you again with us in the following seminars.